Good evening, everybody. How are y'all doing tonight? Good. We're really excited to have you here. Welcome both to our on-site audience and, of course, our fabulous audience joining us online. Bear with me. My name is Sarah, and I'm really happy to welcome you to yet another iteration of our Café Sci program. Café Sci is presented by PPG. It is also, of course, supported by good people like yourselves. Thank you to everyone who donates to support this program. If you are unable to donate but would still like to support Café Sci, the most helpful thing you can do is invite your friends to join us in the audience. Café Sci always takes place on the first Monday of each month, and you can register for next month's speaker at carnegiesciencecenter.org. Before I introduce tonight's speaker, I'll just remind everyone that there will be a Q&A after the talk. For our on-site audience, please raise your hand, and a member of our staff will bring a microphone around to you. Please do wait for the mic and speak into it. Online audiences, you can ask questions by using the Q&A button at the bottom of your Zoom window. You can enter your questions at any time during the presentation, but we'll save all questions for the end. It's my pleasure to introduce tonight's speaker, Dr. Chandan K. Sen. Dr. Sen is a distinguished figure in regenerative medicine and wound care technology. He's an elected fellow of the National Academy of Inventors. Dr. Sen is a university-endowed professor of surgery and directs the McGowan Institute for Regenerative Medicine at University of Pittsburgh. He also serves as the Associate Vice Chancellor for Life Sciences, Innovation, and Commercialization at the University of Pittsburgh. Dr. Sen's leadership focuses on advancing healthcare research vital to Pittsburgh's future, fostering collaborations with local and national partners, and facilitating the translation of scientific discoveries into practical applications for patients. The emergent field of regenerative medicine has been assigned a separate RMAT designation by the US FDA. This multi-billion healthcare market is set to rise sharply, and Dr. Sen's efforts are directed at establishing Pittsburgh as a key leader in this new domain. With a career spanning over 2 decades, Dr. Sen's research has been instrumental in developing innovative solutions for tissue injury, regeneration, and infection management. Notable breakthroughs include pioneering technologies such as tissue nanotransfection and electroceutical infection management. His current projects include leading a biomarker study for the NIH-funded Diabetic Foot Consortium and investigating predictive indicators for diabetic foot ulcer recurrence. Dr. Sen's contributions have earned him prestigious awards, including the 2018 Edison Award for Innovation and the 2016 Frost & Sullivan Award for New Product Innovation. With an extensive publication record and leadership roles at institutions like Indiana and Ohio State universities, Dr. Sen continues to drive advancements in regenerative medicine and wound care. Please welcome Dr. Chandan Sen for his presentation on life after death.
Thank you. Thank you. Thank you. Thank you all for finding the time to be here. And I'm here to share with you a story, hopefully you like. And before I do that, I think it's good to trace the root of the story. And, the McGowan Institute for Regenerative Medicine, I have been a director for less than a year, but this has existed for 30 years. And has given many, many products to the market. And if we sort of try to recap healthcare and healthcare interventions in healthcare, in this country, it was in 1906 that we had the first, what we call now, small molecule drug. In 1938, the first medical device. Now, thousands and thousands of those. Thanks to the 2016 Cures Act, the FDA now has a RMAT designation. What does that mean? That means that now you have a regenerative medicine advanced therapeutic track. What that really means is that fast forward, already now you have approximately 100 products in the market already approved through that track. And fast forward a few years, you are going to start to have complex body parts. These are not metals, these are not plastics. These are body parts customized to your specific needs that would be made and plugged into the human body. That very much changes the way healthcare would go. These body parts could be drug-eluting. It will give you the drugs you need in the right time you need. You don't have to be relying. So there'll be a major shift in the healthcare paradigm in front of us. Who rules that way will depend on who all are at the cutting edge. And the business model will change, reimbursement will change, care delivery pattern will change. People are going to be making medications within their own body, so to speak. So it's a very exciting future. When McGowan Institute was founded, so McGowan, for those of you who would recall, in telecommunications, there was a giant company called MCI. And Bill McGowan was the president and CEO of the company and felt the need, was also a transplant patient, felt the need to contribute to the future of transplantation. And somebody advised him that it would be regeneration. And some life forms in the animal kingdom regenerate very well. We don't. And do we have a chance? I think we do. To the extent it can be helpful to living a more functional life. I don't belong to the camp that believes that extending lifespan sort of indefinitely is a major goal. I think in some ways that could be catastrophic given the new generations coming forth, so on and so forth. But whatever time we have that we are living, to be able to live it full is key. To that end, I think that already major sort of paradigm shifts are in play. You now will get tissue engineered skin as a dressing if you were to be burnt. Very interesting. So it's not like A, B, C proteins or growth factors, but the ensemble of products that the body would make would now be made by this dressing and released into the wound. So speaking of regenerating, do we regenerate? Yes, we do. The best example of us regenerating is when you go in utero. Sounds cruel, just an experiment, please bear with me. When you go in utero and cause wound into the growing embryo, when that embryo is born, you can't even tell where the wound was. No scarring. All appendages fine, meaning you have where the hair should be, the hair is there. You actually have to mark the place with a tattoo to be able to know what part did you injure. So interestingly, when we are in the mother's womb, we are able to substantially regenerate. And the moment we are born, that seems to be lost. And the more we grow, the further we seem to depart from perfect repair. Perfect regeneration seems to be more bothersome and itchy and scary and stuff like that. So is there a way of really trying to recapitulate what happened in utero, in the mother's womb, in the adult body? So that's a significant question. Of course, there are many pillars in regenerative medicine, one of which is tissue engineering. So you get a scaffold like you would build a building. You get a scaffold and you populate the scaffold with bricks and cement, in this case with different types of cells and extracellular matrix that glues them together. And guess what? The organ starts functioning. A good example would be you take a heart, you decellularize the heart, you kill all the cells, only the scaffold remains. Now, of course, the heart doesn't function anymore, right? It's dead. Now, you populate that heart with living cells, the right kind, in the right order. The heart starts beating. You put that heart, you transplant that heart into an animal, that animal runs around. So that's one example of tissue engineering. There are many more fancy examples. But the fact that some, there was a time, I don't know, I think it was during the second Bush era, I think you would recall that there was all this noise about using embryonic tissue for stem cell research, and no, you can't use that and all, and there was a ban, and because people were using embryonic tissue, which we know is very capable of regenerating. But soon, after all those bans, we started looking harder in the adult body and found that the adult body too has many stem cell niche. But even without stem cells, so now we are finding out you don't even need stem cells to repair, regenerate. I'm a little bit scared of stem cells because, they do make the cells you want, but then some of them don't listen to you and end up making cancer. And trials have been stopped. So if we can have cells that can repair, regenerate, and not pose those threats, that would be even better. So it looks like now a big part of our program is reliant on using the skin to actually generate other types of organ function and tissue function. And that's that technology, tissue nanotransfections mentioned about. Today, I'm not here to talk about any of those. While you get excited about something, you at some point start moving in a tangent, sort of trying to explore the limits. You depart from your core. And you start exploring in places that you wouldn't expect yourself to be. This story about human death, ushering in new life, is one example of such digression from our core projects. And I'll tell you why it is interesting. I do have a few slides and I'll share with you, but before I pull the slides, they are more sort of scientific data type. I'll explain to you. I want to really get the story as to why we got interested, what is the crux of the finding, and why is it so important, critically important. First of all, much of knowledge in medicine today is obtained from the dead body. We pretend the dead body is a good replacement for the study of the life body. What I'm going to talk about today will prove otherwise. What is very interesting is that in our search for trying to ignite some of the molecular processes that are in play in the fetal body, when we try to ignite that in the adult body, we stumbled upon a very interesting finding. So of course, we can do that to a limited extent. It's very difficult to approach the extent what plays out in the fetus. It's almost a perfect scenario, what you have in the fetus. But I'll show you this story. What we ended up finding was studying the entire transcriptome, meaning all the genes in the body that you have, of like 700 people, that depending on how you die, some of these developmental pathways, these are pathways that play out in the mother's womb, re-emerge after the human dies? Obviously, the question will be to what end? We'll come to that. The point I'm trying to make is, and I'll show you, there's a Hardy scale of death. You may not care, but to make it sort of summarize it, you have slow death, you have fast death. Fast death, trauma, accidents, suicide, fast death. If you undergo fast death, those developmental programs do not come back. If you undergo slow death, they do. After we die. Now, is it worth paying any attention? We'll see. But when I first saw it, we all sort of paused for a while and wondered why Mother Nature would have this as part of plan. We would think everything is now about to perish. So this story actually is based on this central observation. And what exactly does this mean? So as you can tell, therefore, depending on how the person died, because the gene expression make of the tissues are likely to change, just studying the dead, unqualified as an example of the live, doesn't work. So we have to revisit many parts of medicine that rely on data from the dead, because they may be obscuring our line of progress. That's one example. The other example would be, is it true, and this is a question, not a statement, that organs that are to be transplanted, the performance depends on how the person died? Is it possible? If so, today that is not factored in. As long as the organ itself is available, that's good enough. But what if I told you that organ, the gene expression make of that organ, is already primed? Good or bad is another story. By the way, the person died. Central questions. All right, with that, let's move in and see what we may have here. So we have been sitting on these observations for like 5 years and never planned to write a grant. Everybody hates what you're talking about, human death. Nobody likes talking about it. So then somebody came to my office and said, do you have some crazy idea? Because there's a foundation that funds is looking for, they'll fund what they call big questions. So I asked, what's a big question? And they defined, something that nobody else will want to fund. I said, I may have one of those, a few of those. So they said, write up a proposal. And so I wrote up a proposal, and this is only one can go from the university. So this has to be processed by the university. They said nobody else really isn't interested to submit anyway, anything, so you can go. So we went and came back rejected. But when the came, they were giving comments. So apparently they went to highly accomplished scientists, some 5 or 6 of them, and some of them was giving us some major international award. And so I didn't understand the disconnect. So then I received a phone call saying, Dr. Sen, you submitted this scientific proposal. Can a part of this proposal be religion? So I said, well, it could be, but I couldn't lead. He said, are you an atheist? No, I'm not an atheist, but it's just that I'm not qualified to lead. I'm not comfortable leading. If somebody else leads, I could participate. So no, what if you? I said no. So I couldn't really make the necessary changes. Then they came back again after a week or two and said, can you add philosophy? I said, yeah, that I could do. People call me PhD, and that's philosophy, doctor of philosophy, and my philosophy is pretty weak. So in this way, if I could teach myself a philosophy, so that would be a great opportunity. So long story short, they said, no, the reason we didn't fund you is because you were asking too little money. So add the money for the philosophers, and then we'll fund you. So we added the money for the philosophers, and we got funded. So this is the Templeton Foundation, and thanks to the Templeton Foundation, and it qualified, whatever question we were asking qualified to be one of their big questions, and it got sort of funded. But this is not just to me. There is a team George, pathologist, who works in the morgue, helps us with samples from the morgue. And there was a parent project, very well funded, I think in the 9-digit range in the Boston area, called the GTEx project. And that GTEx project was supposed to map gene expression in all the different tissues of the human. If you look at the call, it would imply that it's the living human, but the project was then run by 2 Boston institutions of high repute, and all the samples came from dead people. So I downloaded. Nowadays, you can download those data because these data are there for public databases for sharing. And we process that data, not our own data, other people's data, and we see what I just told you. So based on that, they funded a prospective study, and George was a part of that GTEx project, the original GTEx project, also our project. See, he was sort of a common link. And then we had, Dr. Singh is here, and computer scientists, bioinformaticians, all 3 of them are computer scientists. Nowadays, data sets are so large, you need computer scientists to mine the data across the genome. And then we had 3 philosophers led by Tim O'Connor, very well-known philosopher based in Bloomington, Indiana. But then he brought together philosophers from Australia, from Canada, from UK, Oxford University. So we really had a big team of philosophers, and we all met in Bethesda, from the entire world. All these people are interested in human death, and they found our observation very interesting. And Peter Noble has been working on animals. And so he came too, and we've sort of formed a group where we can critique each other, complement each other, and build a balanced program. The responsibility to lead was in my hand. And so George King from Harvard, a whole range of clinicians, non-clinicians, Sam Parnia from NYU Langone, quite a team. So I'll spare you the detail of these science, but let's talk about the GTEx project. The GTEx project was massively funded so that personnel at the Broad Institute, involving many other related institutes, could work on identifying and mapping quantitative trait loci in human genome and gene expression in human tissue. The way it was written was this would help us understand, for the living human, And there's a silent assumption, though, in the execution of the project, the entire project was run on dead people. So as far as I'm concerned, all of these data are definitely of value, but we have to recognize that they came from people that died of different causes, and therefore the results are not reflective of what you have in a living human. And that's in the interest of rigor. Now, the title of my project, when we were talking about the title, let me see if I had here the title of our project. Do we say here where it is? Yeah, it is called Human Post-Mortem After Death, Tissue Conatus. Tissue conatus means the human dies, but the tissue wants to live. Clearly. And I'll characterize how the tissue wants to live. I'm going to say, who cares? We do care. It's a very interesting twist of biology that would happen. Looks like when we develop in the mother's womb, the way we develop is to have a functioning human. So different tissues will differentiate in a way that they can work with each other towards a final human performance or existence. However, after death, it seems, based on the data we have so far, and the data is still unfolding, it's too early to call it conclusive, just what we have so far, the tissue de-differentiates, meaning if it is a skin, it recognizes it doesn't need to be a skin anymore. It is just interested to live in the most primitive form with the least energy needed and the least complexity. To be a human, it had to differentiate to a much higher level of complexity. It drops down complexity, preserves life, and seems to be wanting to live. Now, the philosophers don't like me using the word wanting. What do you mean by a will of a tissue, want of a tissue? I'm not going to get into that today, so I'm just speaking plain English, but you know what I mean. So just forgive me for that for the time being. This is just colloquial sort of explanation, but will, want of a tissue is not acceptable at certain levels of sophistication. So let's go forward. So conatus is this urge to live, anything that lives, the urge to live. And I believe if there is a conatus instinct in the body, human body, we better know and we better harness that as we plan intervention, medicine, healthcare. We can only harness once we characterize, once we know what exactly it means and how it functions. So why did we start with the skin of all organs? Because skin, fat, bone, cartilage, these are all tissues that can live for a very long time with very little energy. Energy requirement is very low. So what happens is after we die, of course, there's the resting. Some of us live every month on a $500 budget, $5,000 budget, $50,000 budget. So the $50,000, if that drops to 500, is equivalent to death because the deficit is so large, they're out of business. But the low cost organs, they find a way to linger on. So we felt that these were the better tissues to understand what we're trying to understand. Then we can come back to the more difficult ones that have a much higher expectation of life with respect to resource supply. We can come to that later, but let's try to understand what, therefore, that was sort of another reason is if you look at the Nobel Prize in regenerative medicine, induced pluripotency, meaning you take a cell that is not a stem cell and you make it into a stem cell. So when Yamanaka did that, what did he use? He used a skin fibroblast. So it looks like the skin fibroblast, the skin cells, are willing to become one of anything you want them to become. So that's quite fascinating. You know, we are now turning skin cells, I should say, in the skin into firing neurons that will mature into neurons that can then be harvested, put in the damaged brain, recover brain function. So skin is amazing in power. I mean, if we don't know better, all skin is good for is making boots. But then if you know what they have to offer, you can pretty much make any other organ. So Yamanaka, when we started our story of tissue nanotransfection or TNT, if you Google TNT, you will. So there too, skin gave us, so the work was first published in Nature Nanotechnology. We actually developed a chip that touches your skin for 1/10 of a second. 1/10 of a second once, never again. And the skin converts itself. While it touches, genetic factors are pushed into the skin electrophoretically, process called nanoporation. And now the skin, so there is a set of biological reactions that takes the skin into something else, into say blood vessels, into say nerve cells, and many more. So those are some of the reasons why we pick the skin. Now, these are complex, but let me still explain. So that's called a circus plot. What you see is a circle there. And you see those numbers, those are the chromosomes. And if you now compare, like I said, we'd like to know what happens, gene expression profile in the fetus with the adult, where the fetus are so good and the adult are no good. So we see that certain genes, as indicated in red in each chromosome, are higher in the fetus. Certain genes are lower, the green ones here. And of them, the developmental genes are marked in black. And through these gene names, if you plug into the right software, you get to know what pathway. So in this way, you can find the differential expression of genes in the fetal skin versus the adult skin. Just to get a clue for what's going on so different in the fetal skin, that you have such regenerative response, but not in the adult. So I was talking about the Hardy scale. Of course, if you really look at the scale, it's much more sophisticated than what I just said. It is fast, violent, less than 10 minutes, fast, unexpected, less than 1 hour. We clubbed them into one, fast death. Then you have intermediate, 1 to 24 hours, slow death over the days, you know, predictably you're going to die. Then you have ventilator, we left them out. So we took the slow death and the fast death. We had 81 people, 192 people. Okay? And when we started looking at them, this is the fast death, and that's the slow death, and that's your scale. So the more yellowish or brighter it gets, the genes are turned on. And here are a bunch of genes that all are turning on following slow death. Why? Not one, not two. Sets and sets of genes that represent very meaningful pathways, not random, very properly choreographed. We better understand what is going on. That was our take when we first saw this. So it's not a preponderance. You would have thought the preponderance would be of going down after death. Not so. So in fast death, not in slow death. So when I started looking at the literature, this is not our work, Peter Noble's work and other people's work. So some people are also saying similar things in animals, but they don't have the slow death, fast death sort of setup. These are all animals, so it's a different type of a paradigm. And I think things get pretty serious when you talk about human death. Then all of a sudden people's ears are like, are you kidding me? I mean, don't you have something better to study? You should talk. You're going to misguide the society into darkness. And are you really sure what you're talking about? So there's a lot of sensitivity about really looking at these aspects of human death. So we teamed up with some of them, and recently Peter and Alex and Eric Woods and Mike Levine from Boston and Gould from Florida and from Cairo and me, we all put together a new Unraveling the Enigma of Organismal Death. So we sort of put our minds together, trying to show that death itself, about 10 years ago, when you went for funding to the National Institutes of Health, we just said, oh, we're going to research mice or dogs or whatever animals. But recently, a few years ago, became mandatory to say how many female and how many male. Because this thing called sex as a biological variable, that studying a man is not equal to studying a woman, vice versa. And of course, there is voice on both sides, and SABV, sex as a biological variable, got established. Now, obviously, the dead don't have a voice, but the difference that you see between dead and alive is no smaller than what you see in a man and a woman. I'm not saying that we should not be collecting data from the dead. All I'm saying is that you have to qualify that it is from the dead, and then in some cases where possible, tally that what is found in the live. And the contrast between the two, if any, is going to be very informative. And that approach is going to be in the best interest of rigor in science. So we took these developmental genes, the developmental genes, that play out in the mother's womb. And if you look at fast death, you see a whole bunch of them. So these are all genes, so differentially expressed all genes, not just developmental, so all genes. So in fast death and slow death, so these are upregulated in slow death, downregulated in slow death. You can take those genes that move in a given direction and plug them into certain bioinformatic software. And based on the movement of the genes, the biological pathways potentially in play gets lighted up. And then you know how the signals are flowing in the tissue of these tissues. And interestingly, you will see that these developmental genes and survival genes, PI3-AKT, is a strong survival pathway in us. So survival pathway turning on. And what is turning down? Collagen biosynthesis, collagen formation, extracellular matrix organization, collagen chain trimerization, stuff that you need to make a tissue. So it looks like the cell itself wants to survive, but the differentiation characteristics are fading. But the cell itself wants to survive so the PI3-AKT pathway is a major hub in this. And these are all data from the GTEx, and now prospective studies have just concluded, funded by the Templeton, and a new wave of data will now come that is far more sophisticated than this and prospectively done. And how many? 80 patients we did? So we did 80 patients not patients, sorry, you know, dead people. Now, if you look at the skin, you know, I'm sure many of you would have read in biology names of different types of cells. So, for example, in the skin, you would have read keratinocytes and fibroblasts and endothelial cells, all these cells. Now there's a new way of really knowing what cells are. And this way is the computer tells you that cells with comparable transcriptomic make are one cluster. So the computer clusters them as one. So if you really look at the human skin fibroblast, what I thought was one cell type, that is actually 8 different cell types based on this new knowledge of high resolution. So if you now take a look at all the skin cells here, these are the fibroblasts. And if you now take that and blow that up, you will find from 0 to 7, 8 different cells. And now if you read the journals, not the books 10 years, 15, 20 years ago, but now, then the fibroblasts are of the following type: reticular, sheath, papilla, fascia, pericyte, papillary, hypodermal. So we are gaining more traction and resolution of understanding based on large data sets that were not present in the 70s or the 80s or the 90s, were not present. And that's when the 90s, I went to school. So we're not present, coming up now. Interestingly, if you now ask, where is this resilience most reflected? Which cell type? These are the fibroblasts. The other cell types, the epithelial, seem to give up. Rising up are the fibroblasts. And what exactly is asking these cells to seek a path of life and survival would be very interesting to know. And in pursuit of that knowledge, we now have mounted some additional studies. By the way, when you look at the gene, you then have to look at the protein, and it's all backed up by the appropriate expression of protein. So these are not dummy findings. They have been appropriately validated. And I'll spare you the details. So this is a cell chat analysis where cells are talking to each other. So then you want to know who the fibroblast is talking to, such that their path to life can be realized. And what you see here in purple, from here all the way to there, and some here, are fibroblasts. So first of all, they're talking to themselves, but they're also talking to endothelial cells. So these are the endothelial cells. And this is the direction of talk. Arrow means information coming from here, adipose, fat, to endothelial, and fibroblast. So there's a whole lot of cell-cell talking going on in this tissue. Eventually, one emerges victorious in that they can live. And if you give the right conditions, they will live for many weeks. You can take them out, you can make new skin, you can make them into nerve, you can make them into any other cell type you want. But this inherent interest, without anybody doing anything to them, the fact that they would like to bounce back to life is something very intriguing. I think without making this more complex, and I have a few sort of hypothetical, so this is, somebody asked me that, what do you think is happening? Purely conjecture, purely hypothetical. Soon, when more prospective data comes, we'll have better traction. And I believe, based on my understanding, that, you see, the body is a federated system. And when the key axis, well, what is the key axis? Let's, for the sake of argument, say that the brain and the heart is a major center of control that regulates the function of many other organs and tissues. When that axis is in charge, then all these other tissues yield and integrate and function as a collective body. But when we are on our way to die, and that's the significance of slow death, before we die, our tissues, our organs knows that this is not going to work out. Central system is failing. They then pull back from the integration and serve their own survival needs. And in trying to do so, they de-differentiate because the body is no longer of interest. It is their survival that is of interest. And so these are all testable hypotheses. And so the interesting part of this would be, if that is true, even before we die, way before we die, weeks before we die, there will be concrete exchange within the body where the tissues know that we are ready to die. I'm detached. And one can, hypothetically speaking, trace that process of death has already started. It is just to precipitate at a given time when internally the tissues and organs have sensed it and are now taking their steps. But if I just killed myself, all that cannot play out. And it doesn't, purely hypothetical, but testable hypothesis and more data to come, which will clarify. The broader significance I was mentioning, that as we do investigative biomedical research, it would be very important to qualify data from the death, data from the live, compare, contrast, try to understand. If we don't do that, we could be lost in the dark. Interestingly, it uncovers novel regenerative pathways response. So looks like there is instinctive survival pathways without a doctor getting involved, without intervention done. We have to understand that. Can we scale that to more parts of the body? What is it that really is playing out? Would be of interest to learn. This came from our philosophers, sheds light on life as a purpose-driven process where the purpose is not just the life of the organism, but processes beyond. So the question came, what is an individual? You know, one in many, many in one type of a concept. So that's a philosophical angle. And I just mentioned that you could actually mark the onset of death way before death actually precipitates. And interestingly, is it true that organs that were transplanted and the outcomes of the recipients, can that be traced to the type of death? Are slow death versus fast death providing a different long-term transplantation outcome in recipients? Would be, those are very traceable, very doable experiments. So with that, I think we're on to the final slide. So localization of some post-mortem cells, especially those with low cost of living. And I mentioned that those are the low cost of livings are the more resilient, and they are a good model to really follow. Sufficient time to prime itself for death. We just mentioned that, as opposed to a fast death. Uncover novel survival regenerative mechanisms responsible for such cell resilience after human death. Compare and contrast our findings with gene expression data available in the GTEx. So that's the data that all comes from the dead. So with this, I think I'd like to conclude this session. Thank our sponsors. Much of related work is supported by the National Institutes of Health, Department of Defense, but the actual project is primarily funded by the John Templeton Foundation. Thank you for your attention. I would love to take questions. Thank you.
Dr. Sen, thank you so much. Thank you to our audiences, both online and here on site. I'm going to hand one of these mics off to Leonard. If any of our in-person audience has a question, go ahead and put your hand up. Leonard will bring you one. And I'm going to intersperse that with questions from our online audience. Looks like we've got one here.
So would it not benefit your study to be include as subjects people who are already starting to die? So you could track the skin GM. I mean,
Answer is yes. To get those types of studies approved, I mean, because there are serious ethical concerns, a convincing prospective data set will have to be presented to the IRB, because, you know, it's not fun to tell a family that, you know, we're going to enroll somebody because we know they're going to die. No, we have had, you know, it's very sensitive, and the IRB would like to see. So this study that we are now doing, exactly with that thought in mind, did not include such a group. But now that the second data set will arrive, which is the prospective study, then I think that will open up that particular study design. So the next study can include, but there are, I mean, this is really taking a tissue, meaning you're biopsying somebody. So it's not like just the bodily fluid. It is sensitive, yeah. But yeah, very good question. And that question has come from reviewers too, that even for now, that could you include that? What we did instead was we took skin and we just cut it out of the healthy human. None of this is seen there. So instead of taking the skin from the dead, the fast death or slow death, you just take the piece of skin from a healthy human. These developmental pathways don't wake up there. So this is to do with death, that we know. But then the continuum for the dying, and then what actually transpires before they actually die, when this hypothetical process is already on, obviously of outstanding interest. In the current design, that group is missing, that has to be added. Future design. Yes, ma'am?
Wait, I'm going to ask you to use the microphone so our online audience can hear.
It's not my question, but to piggyback on her statement and your statement of, yes, it's very uncomfortable talking to a family about your loved ones approaching death. Have you ever considered going to hospices where the people have presumably already accepted the fact that their time is limited, and getting some referrals from them to do this study? Because this study has, I can see it having such a big impact on transplantation. I mean, my mind's just, I loved your presentation, first of all, so thank you for that. And my mind was just going, like, I had a million questions while you were going on, and I can't remember all of them. But I would love to see a study comparing, I mean, first of all, I make the assumption, and maybe I'm wrong, but I think from what we read and hear, most transplants are done on people that have traumatic deaths. Your data is almost the opposite of what we would think would benefit a transplant patient. And I would be wondering if anybody could track, and they may have this data already, was it a slow death and a fast death? Who suffered rejection the most, which group, or graft versus host, you know, in the stem cell transplants, and all of the impacts this could have. And the data may already be there for that. Do they track how the recipient, the donor died?
No.
They don't track that.
But it is discoverable based on the nature of
Yeah.
data that is already in the database. Should somebody want to, it is discoverable, but that's not the primary, yeah.
Because the transplants, as we know, are coming primarily from traumatic death, I would think the outcomes, if you're searching, if the cells are searching for survival, it may help lessen rejection and rejection drugs that have their own inherent complications. Am I thinking on that right?
Yeah, you are. It's just a question of really setting that up as a study and following up on that. On the question of the hospice, I mean, clearly that's a place that could be useful. But as you know, when you do consenting of humans, the more incapable they are to speak for themselves, the more complex is the process. It's not like they know they yielded. I mean, there's still a process, but that doesn't mean they cannot be studied. They can. It is just that one will have to just go through that due process and getting the right checks and balances.
Thank you.
Thank you. Yes, ma'am.
We're going to bring you a microphone. Don't worry.
I've always thought that the human body is amazing at what it can accomplish. But what I'm puzzled at is why the body does this. Because there's no benefit to the body itself if the heart is stopping and the brain is stopping, and that's what triggers this regenerative process. Is that what you did say that, right?
Yeah.
So the body itself doesn't get any benefit from these cells being able to regenerate. Other people will get benefits by being able to regenerate other organs, but the person itself, the body itself, it's not going to live longer or survive better. So I just wonder why this process is initiated.
Yeah, I mean, obviously a very valid question. You see, I don't know that, every component of the body, see, a cell can live without the body. And here it seems, based on the signals that show de-differentiation of the cell, meaning walking back from being the tissue or organ that would help the body, the cell seems to be more interested in its own survival than the integrated survival of the body. That's how we look at the human as a whole, because of many other factors. We cannot relate to the individual cell, but it would not be too far-fetched to think that in the entire big scheme of nature, that the cells here you see in the dead body are talking to multiple other cells. And through this process, there is one cluster of cells that seem to be emerging, for lack of a better expression, victorious in finding survival pathways to support an extended time of survival.
But it has to be initiated by the loss of the brain and the heart, or the deterioration of the brain and heart for this process to happen.
Yeah, clearly everything in the body is under a central control, really. There is nothing in the body that really, so there's the autonomic nervous system, there's the central nervous system, there are the endocrine system. So everything in the body is pretty integrated. And there comes a time when that integrated system starts to fall apart and is sensed so by the components. At that time, do these components seek their own survival? The question you ask is a very important one. Right now, we don't have the exact answer. As we delve more into it, possibly we know better. Thank you. Yes, sir.
Wonderful talk, and it's really obviously emerging research. But since one of your other hats is in the commercialization phase, what do you think might come out of this in 5, 10, 20 years?
So first and foremost, I'm interested in the observation that there is a natural survival instinct. See, what works well in healthcare, as far as I'm concerned, is not to fight with nature, but to go along with nature and achieve your goal. That's much more sustainable than being in conflict with nature. You could be extending the life for a few weeks or a few months, but it would be a terrible quality. So if we can learn, what is it in nature that has this instinct to survive? Understood that now only a particular cell type, but it is talking to us. It is trying to say something. If we once understand that, potentially it is scalable. Potentially it can be leveraged to much more complex, sophisticated solutions. Potentially, some of these recent papers that are coming up, I don't know if you have seen that paper from Yale on the decapitated pig head. That was sort of, I wouldn't say brought back to life, but much of the function was restored even after 4 hours of decapitation. What I'm trying to say is bits and pieces are emerging. And if you plug them together, I think you could really achieve some really complex solutions. Instead of us trying to go completely synthetic, to be able to understand what nature is intending and trying to do, and designing our solutions around it, I think that's why I have been spending all this time, so that it's not like what we can go and imagine up and then force it on the body. I think you could do that, and some beautiful papers could come out of it, but those would not be sustainable. So I'm quite fascinated that you have these survival signals and whatever competition and all happening within the body. But why would certain tissue and cells live on? And people have been saying that this also happens in other forms, but I'm primarily interested in human, because the type of what we are seeing, this different form of death, that has never been shown in any other animal type. So that's the main understanding. So yes, we have certain solutions that are today going to the market, but the tomorrow will be far more sophisticated and complex. For that, we need to have some really original pieces coming from Mother Nature, and for us to build around it. So that's the thought process.
Thank you very much. I
Thank
really enjoyed your talk. I was just wondering if in the patients who've died a slow death, if the genes that are upregulated are oncogenes disproportionately represented, and if a tissue sample is transplanted into a patient that had a slow death, could these more stem cell like properties have side effects, have repercussions?
So first of all, did you say oncogenes? The first, did you use the word oncogenes? You see, oncogenes are only oncogenes, when you took up the organism. What is an oncogene? An oncogene is a gene that is very capable of, in this case, actually, we were talking about developmental biology, not oncogenes. But even if it were to be oncogenes, an oncogene is a survival gene, unfortunately for the tumor cell, but it is a survival path. Here, we do not see so far any preponderance of cancer signaling, okay? We don't. But even if we did, that would still give us a cue, because cancer doesn't happen when the tissue is dead. You need to have a lot of metabolism, you need to have a lot of energy, cell cycling, cell division, so on and so forth. Then blood flow is brought into you. So yeah, it finally ends up hurting me, the human, but for the purpose of the tissue, it's enhancing and growing the tissue. So far, of all the genome-wide, transcriptome-wide analysis we are doing, no preponderance of cancer signals so far. But if we saw that in the prospective study, we'll pay attention to that. But so far, that has not been the case. Thank you.
We've got a couple of questions from our online audience, Dr. Sen. So Kitty Mama asks, how does conatus differ from a vital force, from vitalism?
Yeah, I wish I could answer that. In this case, we use the word conatus loosely. It should be used with better clarity, where biological life seems to be perpetuating. Now, there may be many other, abstract explanation. It is just that you'd have many disciplines come and look, and I'm sure that the same observation will be interpreted different by different people of different discipline. And yeah, so I don't know how to really respond to that, but good question. Thanks.
Sure. We've got another one here from our
Yeah.
online audience. An anonymous audience member asks, did your research team need approval from an IRB to harvest skin cells from corpses?
No, we don't. We don't.
All right.
Because it is not a human subject.
Because, okay.
It is not considered a human subject. So no. At the same time, we don't collect the PHI. So I don't say that his son and her daughter. So it's just a non-PHI blinded and not a human subject. Yeah. So
Thank you. Anybody else in the audience? Because you've got another one. All right, here you go.
What exactly is it in the human, in a woman's womb that doesn't allow that keeps something from, keeps the baby from scarring? What protects it? And is that reproducible where it could be used with wounds or after maybe abdominal surgery to prevent adhesions or obstructions?
So, I wish I could tell you exactly what it is. I can't. But if you look at, say, wounds in wound care, amniotic membranes, huge business and does well. Amniotic membrane. So obviously, that's a growth bed, a pro-life bed, has a cocktail. Often we think one versus the other, but it's actually a plethora of signals that is required to support growth and repair. So that tells me the story of the amniotic membrane tells me a lot. If you take amniotic fluid, that's very reparative and regenerative, any which way you measure in the lab. So obviously, a lot is going on in developmental biology, but looks like that when after you're born and you're injured, what happens is repair. But what happens in the mother's womb is they can recapitulate the development. So they go back and redevelop that piece that was injured. They don't patch it, it seems to me, based on the biology I read and understand. So it's not at all similar to what you have, in post-birth. You have inflammation. Their inflammation is not that prominent. So it's a different process. Whatever it is, it achieves a very high level of accuracy in what used to be the original tissue that we cannot replicate today in the adult body.
Thank you.
Thank you.
Thank you. We've got another online question here, also from Kitty Mama. Life as a, quote, purpose-driven process and, quote, processes beyond life sound supernatural to me. What is the evolutionary benefit of any cells surviving the organism's death for any length of time?
So if you look at any organism, it is a concerted effort of various tissues and organs working in harmony and towards organismal function, which we then, and in some cases, you have a cognitive component like in humans, but all of that is a summation of a number of activities that happen, which to some extent can be reduced back to the single cell. If you talk about what is living, it is not the extracellular matrix or the collagen, or it is the cell that is living. That is what reproduces, that is what turns over, so on and so forth. So in this case, if you look at, of course, there are a couple of hundred different types of cells in the human body, but the origin of these cells have a common ancestry. So you can trace them back developmentally. So once you know how these cells behave, I think one can take those information, and based on everything else we know about repair, regeneration, and survival, we can enhance our current understanding and then have the right type of recipe to benefit. So in order to get something that actually can be taken to the market, as the other question was about, sometimes you have to veer off and seek some fundamental piece. This is one of those fundamental pieces. Exactly how, in a few years, I can tell you. But I'm confident it will help us. It will illuminate. Because when we see some type of a cue, we are pretty good at building around it. So this is sort of a natural pillar to build around. And I wish I could. Now, with the second study, now that it's just completed, and we are now analyzing the data, I think in a few months, we would know a whole lot more about how this could be harnessed. Today, the idea was to share with you a provocative story and tickle some curiosity. And that's the purpose here. I did not come here posturing that I know the answer. If I did so, I'm sorry, that was not the intent. It's not like, when you think with your fellow people and you share, and these types of thoughts don't necessarily need trained scientists. Different people having different perspective may come up with thoughts that could end up being very productive. So therefore, pitching these and people that are interested is mainly the reason of this. We have many other things where concrete solutions are going to the market and making a difference, and that is not what we picked, because I just wanted a more provocative, something that is sort of in its infancy, but quite thought-provoking. Yeah. Yes.
Yeah, hello. So I'm a volunteer in All of Us research.
All of
and
Us.
Yes, and they, I think, try to follow a large number of people for a long time. And I know you had to watch a lot of videos before you signed all the permissions. In fact, I thought this is almost biased against busy people, but I managed to get through it. But maybe there's a large set of people that if you ask them, many are healthy, maybe they would give you permission ahead of time. And eventually maybe you can get, volunteers that way.
Yeah. So All of Us, is this currently ongoing, or is that already concluded?
Yeah, I think it's still ongoing. I got an email in the past year.
The genomic, yeah, it's currently on. Yeah, no, that's a good thought. We have to see if we can use some of those resources, because many key leaders of that All of Us are at Pitt.
Right, yeah.
Yeah, and we can certainly leverage them as a resource. Thank you.
You're welcome.
Do we have any additional questions from our on-site audience? Can't quite see everybody up there. No? Oh, Kitty Mama Online says, yes, All of Us is an ongoing study.
Yes.
They want to enroll 1 million people.
Mhm.
I am also enrolled in that study. All right, if there are no more questions for the evening, I think we'll go ahead and wrap it up. Let's everybody give Dr. Sen another round of
Thank you.
applause.
Thank you. Thank you for being here. Thank you. Thank you.
That was a wonderful presentation, Dr.
Yes, sir.
Sen. Thank you. I'm just gonna go ahead and remind everybody that this amazing program happens every month. So please join us on site or tune in for next month on July 8th. And, we could not do this without any of you and certainly not with fascinating presenters like Dr. said. Thank you so much, everybody.
Thank you.